Hello, and welcome to Plants and Python. My name is Alejandra Rougon, and I am a professor at the Genomics Department at the National Autonomous University of Mexico, UNAM, campus en León. This is Command Line Lesson 9, Biological Formats. In this lesson, you will learn to recognize the most common biological data formats for storing DNA and protein sequences, and sequencing, mapping, annotation, and alignments data. Most biological data is stored in text files, where DNA bases adenine, guanine, cytosine, and thymine are represented by 4 letters and the unknown bases with an N. They can be upper or lowercase. If you find a mixture of them, it's because the lowercase letters are masked, and they won't be processed the same way as the others by some programs. Proteins can also be represented by a 1-letter code, where unknown residues are represented by an X. DNA and amino acid sequences are usually stored in the FASTA format. In this format, each sequence starts with a greater than character, then the identifier, and in the next line or the next following lines, the sequence. If the sequence is long, it can be divided into different lines, usually of 60 characters each. After the sequence identifier, separated by a space, you can also have a description. You can also start several sequences in the same file, as each record will start with a greater than symbol and the identifier. Next-generation sequencing data can come in various formats. However, the most common format is FASTQ. The other formats are usually converted to FASTQ for their analysis. In the FASTQ format, each record always contains 4 lines. The first line starts with an @ sign followed by the sequence identifier. The contents of the first line vary depending on the sequencing machine and version. However, it contains information about the sequencing run and cluster. The second line contains the sequence or base calls. The third line contains a plus sign, and in some cases it repeats the sequence identifier of line 1. The fourth line contains the quality scores and each base in ASCII format. In paired libraries, we obtain 2 sequences that come from the same fragment and are associated to the same record. If that is the case, the first line will indicate if it is a fragment 1 or the fragment 2. When sequencing a genome, one has to fractionate the DNA, so pieces of the genome get sequenced. The result is a file with millions of sequences that have to be assembled back to form a genome. That task is usually not completely fulfilled. What we get are bigger fragments than those initially sequenced, called contigs, that come from reads that align contiguously, or scaffolds that are bigger fragments that come from paired reads that can align through different contigs. If we already have a reference genome, we can align or map those reads to a reference genome. The most common formats to show the positions at which each read has aligned to in relation to the reference sequence are the SAM format and its compressed binary version, the BAM format. The BAM format cannot be opened directly with a text editor, as it is in binary. The SAM format contains 2 sections. One is the header, which is identified with an @ sign and 2 uppercase letters at the beginning of the line. Below the header is the alignment information section that contains the positions at which each read has aligned to. The header lines may be removed for analysis. The alignment information is divided into main 11 columns. Additionally, some optional tags can be added for more alignment specifications. The most important columns are the read name, a number that represents alignment information called flag, the reference name, the mapping position, the mapping quality, cigar string, which is another code containing mapping information, reference name of the mate in paired libraries, the sequence, and the quality scores. Genome annotation is the process of identifying the location of genes and all of the coding regions in a genome. It is also called structural annotation. Determining what those genes do is called functional annotation. There are several formats for representing the locations of genes in a genome. Annotation formats contain the name of the genes, the positions at which each gene starts and ends, in reference to the genomic sequence, and the positions of the different parts of the gene that we also call features. These can be UTRs, introns, exons, promoters, and so on. Some genes can have various forms or transcripts due to a process called alternative splicing. Here are some examples of different annotation formats. All of them are tabular, in other words, tables. They contain columns that are usually separated by a tab or a space. Annotation formats are flexible. They may vary, since programs may require slight modifications. These are just general examples. Each individual's genome sequence is unique. The differences in the DNA sequences of individuals are ultimately responsible for differences in observable traits, such as eye color or height, as well as for the hidden ones. For instance, they may determine how susceptible or resistant a plant is to a disease or how virulent a specific pathogen is. In humans, there are 0.1% differences between the genome of any two individuals. That means that, out of a 3 billion base sequence, there is roughly 3 million differences between any two individuals. This variation can be due to substitutions, insertions, or deletions. The VCF and BCF formats are used for giving information about sequence variation. After having our genome structurally annotated, we may want to know the functions of each gene, especially if we have found variation that may represent a significant change in the phenotype of an individual. For example, a variation that may represent the loss of function of a disease-resistant gene. The process of finding the function of the genes is called functional annotation, and it involves the aligning of genes or proteins to a database to try to find homologies to already annotated proteins. The results of those alignments can be obtained in a tabular format. Here is an example of an alignment obtained after running a program called BLAST. In this lesson, you have learned to recognize the most common biological data formats for storing DNA and protein sequences, and sequencing mapping, annotation, and alignments data.